Yeah. Okay, well, it's with great pride that I introduce Alexandra, or Allie Clark, my first recruited student here at UCSD 5 years ago, thereabouts. And I knew she was a superstar just reading her application, and then now I found out recently she's affectionately called the rock star in the JDP year after year. Just so productive, tracking roughly 5 publications a year since she started. More than half of those are first or second authored. I think I'll let her introduce herself. She has some slides in there kind of talking a little bit about her background and her training. But what is, I think, most impressive is she came here with very little exposure to neuroimaging, and I think you'll see just how amazing it is, how far she's come with all the different modalities that she's trained up on and what she's proposing to do for her DP5. So without further ado, here's Allie.
You turn this off, yeah.
You have a thing on?
Yeah.
Okay.
All right, so I want to thank all of you for the opportunity to speak in front of you today. I'm really excited to highlight some of the work I've been able to accomplish here at San Diego VA and UCSD as a graduate student, as well as speak to some exciting new research ventures that are on the horizon. All right. So to give you a brief overview of today's presentation, I'm going to provide you with some background on my experience and career goals, talk a little bit about the NIH Early Independence Award. We're all kind of wondering why we're here. This is partially why. It's a recent grant that I've composed and submitted. Also kind of want to talk a little bit or highlight some work that I've accomplished in graduate school and that has really set the stage for this proposal. And then I'm going to describe the background and research plan of the proposal, and then finally solicit any questions or feedback you may have. Okay, so we'll go ahead and get started with my experience. Okay, so I did my undergraduate at the University of Florida, where I work under the mentorship of Dr. Don Bowers. This is kind of where I was first introduced to research, and I did a lot of research initially with Parkinson's patients. After graduating, I then went to the Boston VA, and I worked with 2 distinct clinical populations there. I studied the cognitive and behavioral effects of mild traumatic brain injury and post-traumatic stress disorder, as well as the association between cardiovascular risk factors and dementia in older adults. I then applied to graduate school and came to work with Dr. Lisa Delano Wood here. And over the past several years, my research efforts have really focused on utilizing a variety of different neuroimaging techniques to characterize TBI-related brain changes. However, I've continued to do some work in the aging sphere as well. I'm currently on internship at the San Diego VA here, where I spend 50% of my time on the neuropsych assessment unit and the other 50% of my time in an integrated mental health and primary care setting. So my research efforts over the past several years have resulted in a total of 25 publications. A little over half of these are first or second authors with, and I've written about 7 neuroimaging-focused first author papers. I've also been able to have some source of funding for most of my graduate school, including an F31 fellowship from the National Institute of Neurological Disorders and Stroke, some dissertation funding through the APA Science Directorate, and then a UC-specific fellowship as well. My research accomplishments also include forging critical relationships with outside investigators and leading experts in the field of TBI, 2 of which include Drs. Aaron Bigler and Lisa Wild. These collaborations have really allowed us to participate in national and international research consortiums. The Enigma and Sensi are 2 of them, where we can leverage existing data sets and the unique expertise of our colleagues in both research and grant applications. And I've really kind of learned a lot through these partnerships about harmonizing behavioral and imaging data, as well as applying advanced multivariate statistical methods to large data sets. So my long-term career goals, I'm going to have to do dual here, are to establish an independent program of research at an academic medical center. And I really want this to focus on characterizing the structural and functional brain changes associated with TBI. understanding the temporal course of these brain changes, and any factors that may mediate or moderate the neural response to head trauma. I also really want to kind of try to continue to disentangle the neurological and behavioral consequences of traumatic brain injury and PTSD. I think a lot of us are kind of doing that work, and it's quite difficult. And then I really want to continue to explore mechanisms underlying the poor long-term outcomes and increased risk for dementia we see after someone has a head injury. So my more short-term goals include establishing an independent research lab here at UCSD. And I really want to focus on integrating biofluid and novel neuroimaging markers of blood-brain barrier dysfunction into my studies of mild TBI. I want to continue to develop and foster these critical research collaborations, and then incorporate some of my mechanistic work into intervention efforts. I think some of the things I'm going to talk about today really could be neurobiological targets for interventions or indices for monitoring outcomes. All right, so next I'm just going to kind of switch gears and talk a little bit about the DP5 itself. So this funding mechanism is intended to launch really well-qualified research candidates who possess scientific creativity and technical skills into independent research careers at an earlier stage. It's really about kind of foregoing that traditional postdoctoral training period. Sorry about that. It's funded by the High Risk, High Reward program of the NIH Common Fund, and only 2 proposals can be put forth by any institution. So here at UCSD, I submitted my grant back in, or an LOI back in June, and this was reviewed by the UCSD Office of Research Affairs. I was selected as one to move forward, and have really been working intimately with our department to kind of figure out key details. I think the thing to kind of take home about this award is that the proposal budget is a lot larger than those CDA or K awards. You're kind of able to accomplish a little bit more. And then it allows for about 80% research effort over the 5 years of the award. So this funding mechanism's been around for several years, but its budget has really grown over the past 3 or 4 years. Last year, around 24% of the submitted proposals were funded, and UCSD has had 3 awards over the past 5 years. So this is going to be the first submission through the psychiatry department. So why apply? Why are we here? Well, I have about 10 years of experience that has really afforded me a foundational understanding of critical pathological processes that occur in mild TBI. And I think this will be highlighted as I review my work and hypotheses. I have research collaborations and access to robust data sets that I can leverage as I establish an independent research lab and an academic presence. I have a demonstrated ability to obtain research funding and disseminate these findings in oral and written formats. And then I've really capitalized on the very unique training environment of the JDP to really develop a technical and quantitative skill set that I think will help me function more independently. And then I've developed an independent program of research that translates findings from the animal literature to humans. And this involves a kind of novel vascular hypothesis, so a new way of looking at TBI. And it really capitalizes on new technology available here at UCSD. Okay. So next I'm going to talk a little bit about the work I've accomplished in graduate school, and this has really set the stage for the DP5. But I'm going to start with some general background on head injury. Okay. So TBI has come to be known as the signature wound of service members returning from the wars in the Middle East. And of the 3 million service members that have been deployed since the onset of the wars, about 384,000 individuals have sustained a TBI. This chart here shows that the vast majority of these injuries are mild in nature, although this may actually be a gross underestimation, given many individuals don't actually present to medical providers following injury. So, how do these injuries happen? Well, one mechanism of injury is primarily related to impact or blunt force trauma. So, for example, if one suffers a blow to the head, injury can occur at that actual site of impact, as shown in figure A here. So right here. Alternatively, damage can occur when the brain rebounds from the initial force in the opposite direction, as shown in figure B there. These are those coup, contra coup injuries that we hear a lot about. And then finally, rapid rotational forces can cause stretching or shearing of the parenchymal tissue and other aspects of the central nervous system. So perhaps unique to the military population are blast-related mechanisms of injury. And really what happens is these explosive detonations can result in a non-visible but really highly pressurized blast wave that may interface with, displace, or kind of damage neural tissue itself. So that's that kind of A point in that figure there. However, blast waves can also cause more kind of traditional blunt force mechanisms of injury as well. So these injuries result in a host of troubling physical, affective, and cognitive symptoms. And they're typically worse within the first 3 months of an injury, and we generally expect them to resolve in the vast majority of individuals. However, this whole notion of symptom resolution is highly debated, as there appears to be a kind of miserable minority that does not recover and continues to experience persisting symptoms. So in reality, what we once thought was a miserable minority may in fact actually be a majority. Importantly, these symptoms are thought to kind of contribute to increased rates of disability and unemployment, poor overall quality of life, as well as higher healthcare cost and utilization among veterans with mild TBI. So we understand how these injuries occur, but significant efforts over the past several years have really focused on answering what structural brain changes underlie these poor neurobehavioral outcomes, and then what neuroimaging techniques allow us to best characterize the neural consequences of head trauma. Okay, so before I discuss precise structural consequences, I really want to first orient you to aspects of the central nervous system that we can use, that we can measure with various neuroimaging techniques. So to the left, we have a nerve cell, which is composed primarily of a cell body and an axon. Cell bodies, or the gray matter, generates the nerve impulse that drives behavior, whereas white matter, or the axon, transmits the impulse between these gray matter structures. So damage to both types of tissue may actually result in behavioral impairment. So we commonly use high-resolution anatomical scans, or T1s, to differentiate between gray and white matter. And in the bottom right-hand corner, you can kind of see an example of what a T1 is, although I know you all are very well-versed in neuroimaging techniques. We then use a variety of image processing packages, such as FreeSurfer or FSL, that allow us to quantify and really divide aspects of gray matter and white matter into functionally distinct regions that we can correlate with behavior. So for example, when it comes to gray matter, we can characterize volume, shape, and cortical thickness. Okay. So in graduate school, I've applied some of these techniques that quantify gray matter in examining the behavioral consequences of mild TBI. Fatigue is something I've been really interested in, and fatigue is one of the most common post-concussive symptoms that are reported after mild TBI. And it's a really complex phenomenon because it has both physical and cognitive components to it. Research in other clinical populations has shown that the thalamus may play a pivotal role in the manifestation of fatigue. However, unclear is the extent to which the thalamus may actually be damaged during neurotrauma, and whether whether this damage is associated with fatigue. So in this study, we explored whether neuroimaging markers of thalamic volume and shape, those gray matter markers, were related to subjective inventories of fatigue. So on the left, we have a depiction of the thalamus in blue, and on the right, we have the association between thalamic volume and self-reported symptoms of fatigue. We found that decreased volume in both the left and right thalamus was associated with higher levels of reported fatigue. And importantly, these findings were independent of age, intracranial volume, or post-traumatic stress, symptoms of post-traumatic stress. Although global volume was associated with symptoms of fatigue, we're also really curious in the regional specificity of these findings. So the thalamus is really a critical relay station with a wide network of afferent and efferent white matter projections. So as illustrated by the tiny bits of red, We found that localized areas of atrophy in the anterior and dorsomedial aspects of the thalamus, in particular, were associated with greater levels of fatigue. So importantly, these regions have prominent white matter projections to areas involved in sustaining and initiating behavior. Thus, we think that damage to this network likely results in the manifestation of fatigue. So I'm not going to go too much into this study, but I do want to highlight that we've used other methods of quantifying gray matter in mild TBI. In this particular study, we showed that cortical thinning was associated with poor executive functioning in veterans with both subconcussive blast exposure and mild TBI. So again, we can use some of these techniques to kind of speak to behavioral dysfunction in TBI. So in addition to gray matter, we can also capture distinct characteristics about white matter, and white matter integrity can be captured using both FLAIR and DTI sequences. So I've published some work using FLAIR that I'm not going to kind of go into, but I want to focus on DTI, as this is one of the most widely utilized and sensitive markers of neurotrauma. So again, we know that during neurotrauma, these rotational forces occur, and that stretching or shearing of these delicate axons happens, as seen in this kind of figure here. Axonal shearing has really been difficult to quantify with more traditional MRI and CT methods, as these techniques don't necessarily capture injury at a microstructural level. Nevertheless, DTI has really provided us with some insight into the integrity of individual white matter fiber pathways. Okay, so this is going to be a really quick and dirty explanation of DTI. But essentially, DTI capitalizes on the diffusion of water within tissue to speak to its integrity. So in healthy white matter, water movement is restricted along the orientation of the axon. In other words, it's highly anisotropic as opposed to isotropic. This can be quantified using a metric called fractional anisotropy, which is a scalar between 0 and 1, and 1 is highly anisotropic, whereas 0 represents isotropic or freely diffusing water. So as such, higher FA values are indicative of intact white matter microstructural integrity. On the other hand, when tissue is damaged, the movement of water is less restricted and more isotropic. So demyelination or cell death may alter the integrity of the axon, allowing water to diffuse more freely in different directions. As such, decreased fractional anisotropy is associated with poor white matter microstructural integrity. So we've used DTI to really explore differences in white matter microstructural integrity between veterans with and without a history of TBI. And I'm going to highlight a study here that cements the utility of DTI when behavioral data may be called into question. So as neuropsychological assessment often includes measures of effort, as adequate engagement is a really critical piece in the interpretation of this data. As neuropsychologists, we want to ensure that any impairments we observed are due to suspected neurological injury and are not merely the result of poor effort. So unfortunately, high rates of poor performance on effort measures has been observed in veteran mild TBI samples, and we've had difficulty kind of making diagnostic decisions about some of these behavioral observations. Although poor effort really may complicate our decision making, it doesn't necessarily negate the fact that an individual may indeed have genuine brain damage, and I think it's kind of important for us to remember that sometimes. Thus, in this study, we used DTI to explore whether there are fundamental differences in white matter microstructural integrity between military controls and those with mild TBI who pass or fail effort testing. So in general, we found that those with mild TBI who failed effort testing, and this was a little bit unexpected, and Mark Bondi the other day reminded me of these findings in supervision, we found that those with mild TBI who failed effort testing displayed reduced white matter microstructural integrity relative to controls and those who passed. So this is, like I said, a bit unexpected but interesting. So we don't think that these brain changes by any means are causing poor effort performance or poor performance on these effort measures, but we think it's important to kind of acknowledge that this group may have genuine injuries and that DTI is a pretty useful marker of injury in complicated assessment. So again, I'm not going to go too much into this, but I have published other papers using DTI that have linked white matter microstructural changes to a variety of different post-concussive symptoms. All right, so to summarize, we've established that structural damage to gray and white matter occurs as a result of mild TBI, and that these brain changes have negative behavioral consequences. However, one thing that's really unclear is the extent to which these injuries evolve over time. We don't know if structural changes are static or if they're worsening. However, we do know that behavioral outcomes appear to be worsening over time. So for example, in this study, increased rates of disability and the severity of post-concussive symptoms increased over 5 years in veterans that had blast-related TBI. So in other words, there's this clear worsening of functional outcomes over time. So these are two questions that I've really been thinking about over the past couple of years. I'm really interested, I've been thinking a lot about what explains worsening behavioral outcomes, and is there another critical pathological process that we really haven't considered? So I began looking into the animal literature to kind of help provide me with some insight into these questions. And across many different animal models of TBI, I was struck by the fact that microvascular pathology has consistently been observed, again, across all different animal models of TBI. In particular, there was this kind of broken and thinning vasculature, decreased blood vessel density in impact areas, and then diffuse microhemorrhages. So in other words, there's clear evidence that structural damage to the cerebrovasculature occurs, not just the white matter and head injury. So importantly, the structural damage has negative functional implications. So the presence of microvascular pathology has also been linked to perturbed cerebral blood flow in many animal studies. Unfortunately, the presence of this vascular pathology in human mTBI studies really remains kind of poorly understood. Few human mild TBI studies really come to autopsy, so we can't kind of tease this apart. And we haven't always utilized advanced neuroimaging metrics that may be sensitive to this kind of subtle pathology. So although we cannot speak to kind of the microscopic changes that occur to the vasculature, we can measure the functional consequences of TBI-related vascular damage in vivo. So as you can see here, DTI has been pretty extensively applied to TBI populations. But over to the right of that, you see that really arterial spin labeling has not been. So I'm going to talk a little bit about what ASL is. So ASL is an imaging technique in which arterial water is magnetically labeled and used as an endogenous tracer to measure cerebral blood flow. Because a contrast agent isn't used, ASL has advantages over being non-invasive, cost-effective, easily repeatable, and potentially more sensitive to microvascular perfusion relative to SPECT or PET vascular imaging techniques. And importantly, ASL provides a direct measure of cerebral blood flow in quantifiable units, unlike bold fMRI. Despite its emerging use in the aging literature, it really has not been widely studied in mTBI samples. So most of my recent efforts really have been focused on utilizing ASL in mild TBI. And this also is kind of largely what my F31 was focused on. So this study in particular was one of the first to apply ASL to a military sample. And specifically, we were interested in the associations between structural or white matter and functional or blood flow changes and whether this differed with time. Okay. So results showed that decreased cerebral blood flow was associated with poor white matter microstructural integrity in veterans who were in the chronic phase of injury. So in contrast, this association was not evident in those who were in that post-acute phase of injury. So that dotted line are those people in that chronic phase of injury. These findings suggest that functional cerebral blood flow changes are especially relevant in the chronic phase of injury, However, the behavioral implications of these findings remain a little less clear. So next I want to highlight some preliminary findings from my dissertation data, and I'm working on kind of currently publishing these findings. But indeed, ASL has revealed that veterans with mild TBI show reduced cerebral blood flow in frontal regions, and this is kind of depicted here on this graph on the left-hand side. And moreover, reduced cerebral blood flow of the frontal lobe is associated with poor cognitive performance. So I've covered a lot, so I just kind of want to summarize a couple of key points here before I explain my efforts to kind of really build upon this research with the DP5. So mild TBI is not just a white matter injury, but evidence suggests that vascular pathology may also occur. Vascular damage likely results in cerebral blood flow changes that can be detected using ASL, and the precise temporal course of vascular pathology and cerebral blood flow changes are kind of unknown, but appear to be especially relevant in the chronic phase of injury. So now I'm just going to kind of switch gears and really talk about next steps, talk about how my proposed DP5 builds upon this accomplished work. Okay. So the integrity of the cerebrovasculature in our brain is critical, and I think everyone in this room probably knows that, but I had to put it on there. Cerebrovasculature is involved in establishing homeostasis, regulating cerebral blood flow, and protecting the brain from outside pathogens, hence the term blood-brain barrier. Our vasculature is the crux of the blood-brain barrier. When the vasculature is damaged due to primary injury in mild TBI, the complex achievements of the blood-brain barrier and may be kind of disrupted, and negative pathological processes are likely to occur and go unchecked. So negative functional changes associated with blood-brain barrier breakdown likely persist and worsen with time, and may be a contributing factor in some of these worsening behavioral outcomes that we see. So here's what's hopefully a more kind of clear theoretical model of what I'm talking about. But basically, during TBI, there's physical disruption to the cerebrovasculature itself. This physical disruption initiates secondary pathophysiological processes that may exacerbate that initial damage, or negatively and permanently alter its functional pathways. And some of the ways that the functional pathways are altered are specifically breakdown of the blood-brain barrier and impaired cerebral blood flow. So we know a little bit more about impaired cerebral blood flow and the regulation of cerebral blood flow, but we don't know a lot about kind of the breakdown of the blood-brain barrier within the context of mild TBI. And then again, these functional changes likely contribute to poor and worsening neurobehavioral outcomes, and I do think there's an association between breakdown of the blood-brain barrier and cerebral blood flow, to just kind of clarify. So biofluid or molecular markers have really been used to characterize blood-brain barrier breakdown in humans. Unfortunately, these are kind of indirect molecular markers of pathological processes occurring in the central nervous system. Their utility within the context of chronic mild TBI is really also kind of poorly understood. Most studies that kind of look at biofluid markers look at these within the acute phase of injury, so within the kind of first year. And then a lot of studies that have utilized these also tend to kind of consist of mixed injury severity samples. There are some neuroimaging techniques such as dynamic contrast enhancing MRI that provide more direct assessment of the degradation of the blood-brain barrier, but these are really not widely available, and again involve the use of a radioactive tracer. However, we have a new alternative that's recently been developed and is currently being implemented here at UCSD, and this is diffusion-weighted pseudocontinuous arterial spin labeling. All right, so I'll talk a little bit about the methods. This is a novel two-stage method which differentially labels water in the blood of the microvasculature and tissue compartments, both of which have unique diffusion properties. And it estimates the exchange rate of water across these tissue compartments. It's a two-compartmental model. Given that water diffuses across the blood-brain barrier, an increase in the water exchange rate may actually indicate increased blood-brain barrier permeability. So importantly, the molecular properties of water make it a more ideal candidate in the assessment of blood-brain barrier integrity, as large molecule contrast agents may not actually diffuse across the blood-brain barrier until a more severe stage of dysfunction or pathology has been reached. So this metric may provide kind of be a useful alternative in characterizing the subtle pathological changes that occur within mild neurotrauma. However, really kind of due to this recent development of this technique, diffusion-weighted ASL in clinical populations thus far has really been extremely limited. Okay. So therefore, I've designed a study that really kind of builds upon this vascular hypothesis and focuses on utilizing these novel neuroimaging methods coupled with biofluid markers to better understand the role that vascular changes play in poor behavioral outcomes in chronic mild TBI. So this is kind of a dense slide here, so I'm happy to kind of go a little bit more into this later when you guys have questions. But basically, I wanted to kind of examine the association between chronic mild TBI, important risk factors for blood-brain barrier breakdown and pathology, and then neuroimaging and biofluid markers of blood-brain barrier breakdown in military service members. So relative to controls, we expect that military service members will show increased blood-brain barrier permeability, reduced cerebral blood flow, and elevated biofluid markers of blood-brain barrier breakdown. And then for hypothesis 1B, we expect that repetitive head injury, increased PTSD symptom severity, and loss of consciousness, which is a marker of injury severity in TBI, will also be associated with these factors. Aim 2 is to kind of link these neuroimaging biofluid markers to behavior. So we want to determine whether neuroimaging and biofluid markers of blood-brain barrier breakdown are associated with poor neurobehavioral outcomes in military service members, and then kind of explore the extent to which brain behavior associations may be moderated by these critical risk factors. Oops, sorry guys, I was reading it off my slide. We really expect to see poor behavioral outcomes will be associated with increased blood-brain barrier permeability, reduced cerebral blood flow, and elevated serum markers. And we also expect, again, that some of these risk factors may moderate the associations we established in hypothesis 2A. So we're going to recruit and enlist about 112 OOO veterans between the ages of 18 and 50 for the proposed study. And the study really has kind of three areas. We've got those neuroimaging sequences I've talked a lot about, plasma markers. These plasma markers are markers of endothelial and vascular injury, inflammatory processes, as well as blood-brain barrier permeability. And then we've also got a behavioral assessment. So looking at not only really comprehensively characterizing these head injuries, but conducting neuropsychological assessment, and then having people fill out kind of symptom or questionnaires related to functional and disability assessments. The proposal aligns with the time of the DP5, so it's going to take place over 5 years, and data collection is going to occur throughout the first 4 years of the award. And then other kind of key things or markers that I'm going to try to make sure I complete national and state licensure requirements as well in the first half of it. So to briefly just kind of summarize, mild TBI is associated with vascular injury. Vascular changes may explain poor and worsening behavioral outcomes in chronic mild TBI. We have new technology that kind of allows us to characterize blood-brain barrier breakdown in humans, and then my research accomplishments, technical skills, and research hypotheses have really prepared me well for the DP5. So then I just kind of want to acknowledge my mentor, as well as Tom Liu, and some other kind of key members of my graduate school experience, as well as the proposal.
or is there somebody in charge of this meeting?
Now, did you get introduced, or?
I did.
Oh, who did that? Then you are in charge. If you did, well, seriously, just to get questions.
Okay. I need some exercise, so we'll run around. All right, any questions? Yeah, Annie.
Well, first of all, you did an amazing job, and you set up the bar really high for us, JD Festival. Do you expect any demographics or any specific factors that may put certain people at higher risk for the brain-blood barrier to be broken down? Like, are you expecting to see differences in sexes, or, I don't know, certain education?
Yeah, well, we do know that there are blood flow differences between males and females sometimes, right? And that's a good question about demographics. I kind of think about the military population as its own unique kind of thing that we have to deal with, where a lot of them have histories of trauma and a lot of different risk factors, health factors, that may actually be influencing the blood-brain barrier as well. So I think it's kind of important that we'll characterize that from a kind of medical perspective as well. I don't know if that completely answers your question, Annie, but.
All right.
Great job. You said that one of your long-term goals is to see how these novel biomarkers might suggest different kind of pathways for treatment. So I know that this is preparatory to that, but can you just give us a hint about what areas you think might be, what kinds of treatments you think, novel treatments you think might result based on whatever you find in the DP5?
Yeah, that's like a very complicated question. And I think that whatever we could do to kind of minimize vascular risk burden, as well as inflammation will be a pivotal piece. But when I was thinking about that slide, I was thinking about a conversation I had with Ariel Lang, where we were talking about whether or not these could really be any markers, or whether I think I would see any response to some kind of other interventions, right? So she's doing a lot of mindfulness stuff. Is there some way that, and I can't really speak to the mechanism just yet, but I'm hoping that we can kind of incorporate some of this into some of those interventions and see.
Hi, great job. A couple of questions about the imaging aspects of your proposal.
Of course.
So sorry.
Of course.
So I saw that you had the sort of couple different approaches with the blood flow and the diffusion weighted, and you mentioned the T1. Are you thinking about sort of structural regions that might be more variable across folks in these measures? You're going to do some parcellations, thinking about either gray and white matter, and you should add a 3D too. But we can talk about that later. So I wondered what your thoughts were there.
Yeah, I do think that there certain regions are probably going to be more vulnerable. I expect that impact regions, or even, there's kind of diffuse axonal injury, and I think some of these like microvascular changes are diffuse as well. Mm-hmm. But interestingly, with a lot of our kind of blood flow work, we see that there is this kind of temporal and frontal predilection that really kind of maps onto what we're thinking about when it comes to models of TBI.
Sure, and be interesting to think about how the vasculature maps on.
Yeah.
There's certain areas that may be particularly more susceptible, so it's really going to be interesting.
Mm-hmm. And we should talk.
All right. Yes, Carrie.
So this is really interesting to me, especially as we're always, with our brain tumor patients, trying to move from less invasive to, from more invasive to less invasive. Yeah, hopefully not the other way. But I haven't looked at the literature on this, and I'm just very curious. Are there studies in TBI that have looked at the association between K-trans and ASL perfusion estimates, like how comparable they are?
Not that I don't know, Carrie. I don't know the answer to that.
And that's a tough one. It's kind of a personal interest one, because we'd love to be able to replace our DCE or a DSC with ASL, but there's not a whole lot of literature that I found out there comparing it, and wondered if you had come across any.
Yeah, there is. I think it has been applied in some brain tumor patients. And then, of course, because I'm not super interested in that, no offense. I just kind of glossed over it, but I have a couple papers that I can send your way, for sure.
But not anything in TBI.
Not
that would
anything
speak
in
to
mild TBI,
in
no.
terms of how the permeability estimates compare across the device.
No, actually, hold on, I'm thinking about that. I think not with diffusion-weighted ASL, and that's kind of, I'm thinking about the bias of that lens, but there is some stuff with DCE, MRI, and sports concussion samples. So I think there was a recent NFL study, and it was just like a little abstract that I saw a couple weeks ago, where they had looked at this and they had seen that there was, as detected by DCE MRI, blood-brain barrier breakdown in people that had had repetitive head injuries over the course of the season.
Right, any other questions? Yes, Susan.
Great talk and great turnout, by the way. I haven't seen a job talk this well attended in a long time, so well done. I was just wondering about substance use. I do substance use research. We've done a little ASL and seen changes in people's, when they become abstinent, with changes in their ASL over time. And I was wondering if you see a lot of substance use in your TBI population, and if so, in your studies, would you have a minimum duration of abstinence or anything like that?
Yeah, so primarily, I should say, with Lisa's studies, we've really been kind of making sure that no one is currently dependent or abusing alcohol when we're recruiting them. And I think I'll probably take a somewhat similar approach. I think it's important for us to absolutely characterize current use as well as past use, and then to kind of be able to think about, okay, how might this be influencing some of the things that we're seeing? Ooh, sorry. And yeah.
But one problem is we're trying to get a pure sample, clean sample, and so many of the potential participants who come through are using often marijuana every night.
Yeah.
They're drinking pretty heavily, the PTSD. So what is it? We screen, 10 out of 11 out of the, so.
Yeah, we exclude a lot of
the
people.
good news is we're looking at the effect, we feel confident, we're looking at the effects of TBI. But we're not able to really characterize that because of just how much we screen. So, but it's important, and there are folks like Karen Hansen-Bondi who's really interested in that. She's published a little bit using our data.
So I'm kind of interested in the the blood-brain barrier breakdown. The acute TBI literature talks about that quite a lot, but I haven't really thought deeply about how they, I assume were measuring chemicals and not doing ASL. Is there an ASL literature in the acute TBI area? And the question would be, it may be then that there is a blood-brain barrier breakdown acutely that doesn't resolve rather than, increases over time. And do you have any thoughts about that?
Yeah, absolutely. So there is some, pretty much ASL has not kind of been widely applied to military TBI samples, but it has been applied to kind of sports concussion samples. So we can kind of think about it within the context of that. And then actually ASL was recently endorsed. I think there's a paper out this past month in Brain Imaging and Behavior for the NCAA CARE Consortium. So they're showing that ASL is detecting, or that ASL is useful in detecting blood flow differences in a sports concussion sample. Really, there's mixed literature on whether this is kind of increased blood flow perfusion or decreased, and I didn't kind of get into that within the context of this study. But that's another thing that we kind of need to figure out, is what direction it's in and why it's happening in that particular way. I do think that there could be blood-brain barrier breakdown could happen acutely, and that there could be some people that actually repair, and then there could be a subset of people that just kind of continue along this pathway, and then maybe that's these chronic mild TBI people, these people with these worsening behavioral outcomes.
And do you know, Allie, in what other populations the blood-brain barrier has been examined, sort of using similar sequences that you're proposing?
Yeah, so DCE MRI has been a little bit more widely utilized, but when it comes to the diffusion-weighted ASL sequence, I think only like obstructive sleep apnea sample and tumor samples is the only other kind of clinical populations that I've seen it in. And it's kind of, I think it was developed at Wayne State, And like I said, we're kind of just beginning to implement it here, and we're still very much in the developmental phase.
Optimizing it currently with
yeah.
Tom Lu. Okay. Yeah, Amy.
Thanks, Allie. My question was, if I saw your hypothesis correctly, you were speculating that PTSD might moderate that brain behavior, blood-brain barrier, rather, relationship. What would be the mechanism of PTSD moderating that?
Yeah, so I think there's been a lot of, we have somebody kind of studying here at UCSD the relationship between neuroinflammation and PTSD, and I think that having PTSD and having it over a prolonged period of time and greater symptom severity, that might be kind of causing that unchecked neuroinflammation. And there's a lot of evidence to suggest that unchecked neuroinflammation is bad for the blood-brain barrier.
Just one more comment to follow up on the different disorders that the diffusion-weighted ASL has been looking at. We've been, I already spoke with you.
Oh, HIV, yeah.
So we were talking with Tom Lu and Greg Brown about using it in the HIV infection, and Jenny Udicello has just gotten an award that we're going to start, so we'll be helping push the development of that with you, because HIV is a very appropriate place for us to look at that as well.
Mm-hmm. And also kind of speaks to that neuroinflammation piece with PTSD, right?
Yep. So we're going to
That's awesome. Good news. Yeah, Igor.
One question. I'm not thoroughly familiar with this area, but S100 beta goes up and down depending on how much brain injury there is and how much leakage there is. Is that true? It's not a totally linear relationship?
Yes, and I'm not as kind of well-versed in some of the kind of temporal pieces to the actual biofluid markers, Dr. Grant. I wish that I was, to be completely honest with you, and I think that I've had some recent conversations with Susie Hong that's been helpful, as well as some members of the CSAM biofluid core. So really kind of when I was trying to isolate or pick some of these biomarkers, I was thinking about, okay, what has been used and what's been sensitive to mild TBI in the past, but absolutely the temporal course and pathway of these things, just as I talked about in the talk, is really important.
I guess my point was more, and again, this is really a question because I don't know this area in detail, but how much is this marker a marker of acute opening, if you will, of the blood-brain barrier versus persistent small leakage and so forth? Mm-hmm. That's one question. And the other is there any reason to measure something related to a biomarker of neuronal injury itself? Now, you're, of course, going to have lots of imaging, so that's a very good biomarker. But have you thought about that as well? Neuron-specific ELAs or something of that nature? Mm-hmm.
Yeah, no, I think we'll kind of need to take a closer look at some of those serum markers and try to kind of figure that out a little bit more.
Great project, though. Oh, and the other thing is, I didn't hear Susan's question fully because I got distracted. But
I think I got distracted too, Susan, so sorry.
I assume you mentioned tobacco as a substance.
Actually, we haven't looked at that, but tobacco is obviously
because that's going to affect
sure.
a lot of these things, and we often forget tobacco as a drug.
For sure.
And I assume military may be overrepresented with tobacco use.
Yeah.
I don't remember we looked at that, but what they we could take another look. I don't remember exactly whether they're hugely high, but they do like to chew and use snuff rather than the smoke. Mm-hmm. For sure.
Well, so it could affect the endothelial markers of that nature.
Mm-hmm. Absolutely.
I think, Alan, you looked at this a little bit with your white matter lesion paper, and I think you were surprised, right, about just the rates of cigarette smoking being much lower than you anticipated?
Yes, I was actually looking at nicotine use in our sample as it relates to blood flow, and because we screen out a lot of things, I was actually surprised that we didn't have a lot of use in our particular sample, but I think we just inherently tend to kind of weed a lot of these people out.
Right. Yeah, we were excited to look at that, and then we kind of couldn't with our sample. Any other questions?
course, can I make my usual marijuana? Of course, if they use marijuana, that will close down any blood-brain barrier leakage they might have had due to the TBI. So you better look at that too.
That's good news.
Talk to you later.
Any other questions? A little bit more time. All right, well, good job, Allie. Congratulations.